Okay, hi everyone. I just got a new computer setup in my office, and so with any new setup, I'm trying to have to relearn how to get everything working for our lecture and with Zoom. So hopefully this is working okay. If there's any problems with it, feel free to send me a message in the chat. I'll have the little chat box open. Okay, so let's talk about photosynthesis, right? And how the management of energy and the creation of ATPs are done in plants. So we have previously been talking about how all life on Earth, nearly all organisms on Earth, utilize glycolysis as the kind of starting point of taking glucose and breaking the glucose down to be able to get some ATP out of it, right? So the energy that's in the glucose needs to be converted into ATP, which is the energy currency that the different molecular pathways inside the cell are geared up to use. So we've looked at the breakdown of glucose, just starting with if you have glucose in the first place as the raw fuel. So that's why it made sense, since we're talking about glycolysis, to then go ahead and talk about aerobic respiration, right, which is what life evolved to add on. Right? In addition to glycolysis, once there was plenty of oxygen in the atmosphere, and as we learned last time, the role of the oxygen is to serve as the final electron acceptors in the electron transport chain, right? So as a quick review, glycolysis in our cells happens in the cytoplasm. We get a couple of molecules of ATP out of it, and we convert it into pyruvate, right? Just like every other organism that does glycolysis as well, you end up with pyruvate, right? But in our cells, because we have access to oxygen, that pyruvate can then be converted into acetyl-CoA, and then the acetyl-CoA gets fed into the citric acid cycle, right? And remember, the purpose of the citric acid cycle is not necessarily to generate many more ATPs from the cycle itself. The cycle is more about breaking the carbon-carbon bonds, right, and converting those electrons into free electrons that can then be carried away from the citric acid cycle by these coenzyme baskets, like the nicotinamide adenine dinucleotide, that's the NAD+, that becomes NADH once it's carrying a pair of electrons, and the flavin adenine dinucleotide, or FAD, right, that becomes FADH2 when it gets loaded with a pair of electrons. And then we learned last lecture how those electron-carrying baskets, these coenzymes, then empty the electrons into the electron transport chain, which is that series of large protein complexes in the inner membrane of the mitochondria in our cells, that then uses the flow of electrons through these big protein complexes to pump protons across the inner mitochondrial space, setting up a very strong proton gradient inside the mitochondria. That is then allowed, those protons are then allowed to flow back downhill through the mitochondrial membrane through special protein complexes like the F-type ATPases, which serve as ATP synthase complexes, right? The flow of electrons through those going back downhill, because not the flow of electrons, I'm sorry, the flow of protons going back downhill, right, powers the spinning or the turning of these remarkable ATP synthase complexes, generating with each turn 3 ATPs, right? And then the net result at the end is, even though you started off only making 2 ATPs from glycolysis, right, with the help of aerobic respiration added onto it, you end up actually making a total of 38 ATPs from that starting glucose, right? Which is a tremendous improvement. And that is how our cells in our body are able to power all of the different functions inside of them, right, using that ATP. So that assumes, right, that we're starting with glucose, But where does the glucose come from? Right? In our bodies, the glucose comes from the food that we eat. Right? And where does the glucose come from that's in that food? Well, this is a discussion of the food chain, right? You go far enough back in the food chain and you realize, well, it all starts with plants, right? Plants that are the photosynthetic organisms that are taking the energy from the sun, right, and putting together those glucose molecules to start off with. The plants do it not to help us. The plants do it because then the glucose molecules can be distributed inside the plant. And then each of the plant cells can then take the glucose and do glycolysis and aerobic respiration to get the ATPs back out of it, right? But, we as animals, we mooch or steal from the plants. We eat the plants in order to get that glucose that the plants made for themselves, right? And that's how, we get our glucose. And that's the story of life on Earth, right? You have the organisms that can make their own glucose through photosynthetic means, and then you have the organisms that basically take from those photosynthetic organisms, right? So that's kind of the balance. But it does balance out, because just as the plants are making glucose from carbon dioxide and water and releasing oxygen as a waste product, right? We eat the glucose and we breathe in the oxygen that the plants have made in order to do our aerobic respiration, and we release carbon dioxide, right, as part of the citric acid cycle and other steps in aerobic respiration that the plants then use, right? So that's the cycle of life. So let's talk about photosynthesis then. How do plants take the energy from the sun and make the glucose in the first place? So this is a picture of probably one of my favorite places to visit. And this is a pond. This is Cutler Pond, which is located in Boston, or actually it's in the suburbs of Boston. And this pond is about, like 5 minutes from the house that I grew up in. And as a little boy, I used to go here with my classmates as far for field trips, right? So we'd learn about nature, walk on the trails through the woods, see the fish, frogs, things like that, catch poison ivy, all that great stuff that I'm sure the teachers loved. But I also have very fond memories of my dad taking me to Cutler Pond to go fishing, especially during the summer, right? So during the summer, Cutler Pond looks like this. It's very green, it's very lush, and there's lots of fish. And so as a little boy, I used to fish with my dad, and I'd catch these catfish, right? And these catfish, they're like all muscle, right? So they'd fight and fight. And as a kid, I was convinced that I caught a whale, right? And I'd be reeling this thing in, kind of freaking out, but very excited. And then we get this catfish. And then my dad would help me, clean it up, and we'd take it home and we'd fry it up. And it was really yummy. So I have lots of fond memories of this place. So this is what it looks like in the summer. But as with most areas in New England, it looks very different in the fall. So in the fall, it looks dramatically different. It looks something like this, right? So all the leaves have changed to these amazing colors, bright oranges, reds, yellows. And part of the reason why I love this pond so much is I have all these memories of different seasons visiting this pond, right? So as an adult, my wife and I, we lived in Boston for several years as I was doing my postdoctoral studies. And we'd come to this pond and we'd go for walks together, right? And oftentimes I would just come by myself because I like to go running. So I'd go and run this loop. It's a short loop. It's only maybe a 2-mile loop around this pond. But I loved the fact as an adult, as I'd be running through these different trees and things like that in the different seasons, it would make me think about all the time that has passed since I grew up there. Because for those of you who are familiar with trees on the East Coast, right, they are deciduous. That means they change colors through the different parts of the year, turning to these bright colors in the fall and eventually losing their leaves in the winter. Right? And so all through that time, the leaves are an important part of the photosynthetic functions of these trees and plants. Right? So let's talk about photosynthesis. So photosynthesis is all about taking energy from the sun, which we have an abundance of, right? Because ever since the beginning of life on Earth, 3.5 billion years ago, there's been plenty of sunshine, right? And that's a lot of energy. So it's all about capturing that energy and converting it into a chemical form, right? Which is, of course, ATP. Right? Then taking that ATP and turning that ATP into carbohydrates so that the carbohydrates, which then represent the energy from the ATP, which itself represents the energy that was captured from the sun, can then be sort of shipped around inside of the plant, right, to the different tissues and different cells of the plants that themselves don't do photosynthesis, right? Because remember, the leaves of a plant do photosynthesis, right? But things like the stem or the trunk of a tree or the roots don't do photosynthesis, right? So how do they get their energy? Well, they get it in the form of the sugars that are made inside the leaves and then move down to other parts of the plants. And of course, as sugar, it can be stored for later use, right? Just think about things like root vegetables, right? That store the sugar either as sugar or as starch, right? So this photosynthetic process then is in two parts. The first part we call energy transduction. So this means the capture of solar energy and conversion into ATP. The second step then is to take the ATP that was made and make carbohydrates like sugars, right? So we call that carbon assimilation. Right? Notice, we always think about photosynthesis as sort of just one sort of generic cartoon that just has things like carbon dioxide, water coming in, and then sugar coming out, right? So now we're going to explore this in a little bit more depth. And the first realization is it's actually in two parts. The water is important for the first part, that is the energy transduction part, right? And the carbon dioxide is important for the second part, the carbon assimilation, right? Already you might get a sense of how this is going to work. Energy and water are used to make ATPs. And one of the byproducts of this is oxygen. Right? On the other hand, once you have ATPs available, you can then use those ATPs to take the carbon from carbon dioxide and rearrange bonds to make sugars. So that's why the overall net equation is light plus 6 molecules of water plus 6 molecules of carbon dioxide give you 6 molecules of oxygen and a molecule of glucose. So what I want you to notice here is that the H2O and the CO2 coming into these cartoons are involved at different parts. And I also want you to think about, again, how photosynthesis, when we think about it this way, is really the reverse of glucose oxidation, right? So we've seen before cartoons where we had a cartoon that on one side, it was the breakdown of glucose, giving us our minus 686 kcals per mole of energy. And on the other side of the cartoon, we had something showing it going the other way, right? And that it takes 686 kcals of energy to put carbon dioxide and water together to make glucose, right? So here is glucose oxidation. It is basically the reverse of photosynthesis. And I think that's wonderful to think about it that way, right? Because as I said, a few minutes ago, life on Earth is all about that balance. Organisms that make sugar, organisms that eat the organisms that make sugar to get that sugar, right? And then we fall into that second group. We release carbon dioxide that is used by those photosynthetic organisms to make the sugar in the first place. Right? Inside of a plant cell, so here is a maple leaf. So, I like maple leaves because those are the types of trees that I grew up with on the East Coast that change colors so nicely, right? If you were to zoom in and look at an individual cell, you would see these little green sort of specks inside of it, right? So these green structures, if you were to zoom in, these are the thylakoids, all right? So, or chloroplasts, I should say, right? The whole structure is known as a chloroplast. Inside the chloroplast, if you were to cut it in cross-section, you would see that it's made of many of these stacks of like little pancake structures. These are the thylakoids. And so this chloroplast, right, has a lot of similarities to the structure of mitochondria. That's not a coincidence, right? Because remember we discussed how eukaryotic cells, like our cells, that are made of many different organelles inside of it, in ancient primordial times, when everything was just prokaryotic cells, you probably had large prokaryotic cells that took smaller prokaryotic organisms into it, right? Not because they're eating them, but just because their membranes probably surrounded them. And both organisms benefited, right? The small organism, the small prokaryote, has a nice place to kind of hang out now where it's protected, being surrounded by the larger prokaryote. And then all the metabolic kind of products that the smaller prokaryote was making then gets taken advantage of by the large prokaryote that's surrounding it. And that is most likely how eukaryotic cells formed, right? So over eons of evolution, they ceased functioning as separate organisms, right? And they just became eukaryotic cells. So that means, just like with mitochondria, that fits that kind of story, chloroplasts most likely fit that same story as well. And we can see that because very primitive single-celled prokaryotic photosynthetic organisms like cyanobacteria, so this is cyanobacteria right here. This is just a single prokaryotic bacteria that's photosynthetic, right? They don't have separate structures inside of them that are membrane bound like chloroplasts, right? They themselves are like a chloroplast. In other words, they're filled with chlorophyll, right? And some very rudimentary sort of like membrane structures that allow things to be sort of pumped back and forth or moved back and forth within them. So these ancient cyanobacteria most likely became part of eukaryotic cells. They became the chloroplasts of plant cells, right? Because they evolved this symbiotic relationship with the larger cell that now surrounded them. Okay. This cartoon looks a lot like the cartoon that we looked at before of the mitochondria, right? So that's why I'm trying to emphasize to you that there are many parallels between the chloroplast in plants, right, and the mitochondria that are in plants, right, and the mitochondria that are in our cells as well. So let me dispel something really quickly, right? I think a lot of students get confused and think that plant cells have chloroplasts but no mitochondria, and that our cells have mitochondria but no chloroplasts, right? So of course that's true that our cells don't have chloroplasts, right? Because we're not photosynthetic, but plant cells most definitely have both chloroplasts and mitochondria, right? And I think I've told you enough to already make sense of why. The chloroplasts are where ATP is made, right, so that you can make sugars. But then the sugars have to be moved around, right, to transport it to other parts of the plant, and plant cells that don't photosynthesize themselves, right, that maybe don't have chloroplasts in them, have to take that sugar in, and they have to do aerobic respiration, right? So they need to have mitochondria. So plant cells have both chloroplasts and mitochondria. So this cartoon then looks a lot like that busy cartoon when we looked at the mitochondria, but let's see what's going on that's different inside of a chloroplast. Right? So inside of the chloroplast, you have those stacks of pancakes. These are the thylakoid sacs. And you can see, as it turns out, the stack is actually a continuous space. Right? That's kind of like folded up like this, and it gives you a lot more sort of surface area to work with. You'll see that light is going to be coming down and penetrating the leaf and getting into this plant cell that's in the leaf and getting into the chloroplast. So this energy is light energy, and it's going to be received by these protein complexes that are part of the membrane here. And so for those of you who are perceptive, you might say, yeah, this kind of looks like in the mitochondria, the cartoons that show the electron transport chain proteins, right? Those big brown blobs in the mitochondrial cartoons that we saw. And we said those big brown blobs in the mitochondria, their purpose was to take electrons and allow electrons to pass through them as they're being passed from little protein subunit to protein subunit, and pump protons across the membrane of the mitochondria, right? Hey, so surprise, surprise, that's what's happening here too. Right? In this case, electrons are also flowing through these big protein complexes, right? But notice, where are the electrons coming from? In the mitochondria, the electrons were coming from all of those electron-carrying baskets, those coenzymes that were being filled up with electrons in like the citric acid cycle, right? And then those baskets were emptied into the big brown blobs, those big protein complexes of the mitochondrial ETS. And those electrons would then flow through. Here, you don't have those baskets dumping the electrons in. The electrons are coming from water. Remember, oxygen is a very greedy electron acceptor. It would love to take 2 electrons to fill up its outer orbital, its valence orbital, right? And along with those 2 electrons came what? The 2 protons that were bound to those electrons. Remember, I told you, electrons don't just float around by themselves, right? They're always associated with a proton. And once you have a proton and electron, I mean, that's hydrogen, right? So water then represents the oxygen that received 2 electrons with the 2 protons that came with it, and that makes H2O. So in the thylakoid stack here, the water is broken and the 2 electrons are taken off, right? And those electrons then represent the electrons that are then going to flow through this electron transport proton pumping system in the chloroplast, in the thylakoid here, right? And as a result, when you break the water and take the 2 electrons off, the 2 protons then are freed up, right? And you get oxygen released, right? So this is where oxygen is released by plants as a waste product, right? Just from the breaking of water to start off this electron transport chain, right? And what's going to push these electrons through these proteins, right? In this case, we're not talking about a big range of prosthetic groups, because remember, in the mitochondrial ETS, each of those big protein complexes are made up of different proteins that all had different, I call them baseball gloves, right? Different chemical groups that were very good at taking electrons and passing them through there. And those different groups were arranged, those baseball gloves were arranged so that the ones at the start would much rather pass the electrons on than keep them, whereas the next one down the line is happier taking the electrons, right, rather than passing it on, until you eventually got to the end where you got the baseball gloves and eventually the oxygen that is happy to take the electrons and keep it, right, in the form of water, right? So here, you don't have a whole bunch of prosthetic groups, right? So this is going to be powered then by the light energy. In essence, the light energy is going to be the thing that kicks or moves the electrons through this to make the proton gradient, right? From this point on, the story is similar to the mitochondria, because what is this proton gradient used for then? These protons would love to flow back downhill, right, out of this thylakoid lumen. And then here is where you have those ATP synthase pumps, just like in the mitochondria, right? So you make a whole bunch of ATPs over here, right? So for today, let's focus on what's happening on this upper part of the figure. Okay. So the light is being captured in these protein complexes by special molecules, right, that essentially are like the one main type of prosthetic group used to kind of deal with these electrons, right, in photosynthesis. And among these are the chlorophylls. So this is a structure of chlorophyll A and chlorophyll B. So you can see, just like the baseball gloves or prosthetic groups that we talked about that are used by the mitochondrial electron transport chain, right? Chlorophyll is itself one of these prosthetic groups, right? So it has this big structure up here with a magnesium in the middle, right? So magnesium is the ion, the metal ion that's used to make chlorophyll. And it has this long kind of tail, this kind of hydrocarbon tail. So here's the structure of it, right? It kind of looks like, I think it looks like a net that would use to catch bugs, or you take it to the tide pools, right? And you're fishing around in the rocks trying to frantically catch fish. So I think that's kind of an appropriate analogy, because after all, this is a net. This net is catching something, right? But what is it catching? It is catching photons of light that are raining down on the plant. So the structure of this part of the chlorophyll, you can see all these double bonds, right? All of these double bonds can kind of flip around and change their location within this kind of like net-like structure, right? So as photons of light, as energy hits this structure, it causes those double bonds to kind of alternate, right? Sort of like an effect of like a something hitting something, and you have vibrations or ripples coming out of it, right? And that energy of the electrons being kind of bounced around like that is what's going to be transmitted on, right, as part of powering the electron transport chain. In addition to chlorophyll A and B, so chlorophyll A and B only differs by the whatever R group is attached here. So in chlorophyll A, it's a methyl group, and chlorophyll B, right, you can see it has this group over here, right? There are other types of pigments, so we call these pigments because they have a color, right, such as phycoerythrins, phycocyanins, beta-carotene, lutein, right? And they're all different colors. So you can see here, they're different colors because their individual structures are particularly tuned in to be able to capture certain wavelengths of light better than other wavelengths. So chlorophyll A and B, the one that we looked at, this big structure here, on this figure on the left, what it's looking at is on the x-axis, you have the wavelengths of light, right? So in this case, going from 300 nanometer wavelengths of light to 1000 nanometer wavelengths of light. And on the y-axis, what it's showing is how sensitive, how good at absorbing these particular wavelengths of light are these different pigment molecules. So chlorophyll A is in light green, chlorophyll B is in dark green. So you can see the light green is over here, right? There's a peak over here at maybe like 420 or so, right? And then it's not very good at absorbing wavelengths of light from this point here, maybe about 420, 450, up to maybe like around 600 nanometers, right? But then beyond 600 nanometers, once the wavelength of light that's hitting it is wider than 600 nanometers, right, it is sensitive to absorbing that wavelength again, up to around 700, right? And so the chlorophyll B, the darker green, similar, right? It has highest sensitivity down here in the maybe 450 nanometer range. It's very insensitive, cannot absorb wavelengths in this middle part, 500 to 600, but then it's sensitive again at maybe around 650, right? Notice that the other pigments are different. The beta-carotene in orange, right, is very sensitive from 400 up to beyond 500, at which point it cannot absorb anything bigger wavelengths than that, right? Or the phycocyanin, for example, in blue, right? It cannot absorb anything below 500 nanometers, but it's very sensitive to wavelengths that are bigger than 600. Right? So what does this have to do with color, right? This little rainbow pattern down here reminds us that these different wavelengths of light in this range, 400 to 700, represents the visible spectrum of light, right? Wavelengths in this range are what we see as the different colors. So why is chlorophyll A and B green, right? Why are green leaves green? Because look, they absorb all the wavelengths that are below 500. So they absorb the purples and the blues, and they can absorb the yellows, oranges, and reds, right? So that energy of those wavelengths, when it hits the chlorophyll molecule, right, the energy is just absorbed as, in terms of all these electrons then bouncing around in these double bonds that are then flipping around. But the wavelengths in the middle, including green, right, at around 550, are not absorbed. And so those wavelengths, when it hits the chlorophyll A and B, it just bounces back, right? And when it bounces back, that's what bounces back to our eyes, right? So when we look at a green leaf, it looks green because its chlorophyll A and B is absorbing the other colors in the sunlight, but the green color bounces back, and that's what we see, okay? All right. So let's think of light. What is light, right? So light is energy. I think most of you are familiar with this concept of light being considered both like a particle and also like a wave, right? I think this is what you learn in physics, right? I always thought that was really weird. I didn't understand it. I thought that was a little too Einstein for me or something like that. But let's take it apart and think about it. It actually isn't that hard to comprehend, right? When we think about wavelengths, so in the purples and blue range of light, we can think of that as being higher energy, right? Because the wavelengths are a lot shorter. As you make the wavelengths go wider and wider, up through 500, 650, up to 700, right? They represent lower energy. Now, you might not think that's very intuitive because you might think, wow, a big wave has more energy than teeny tiny waves, right? But no, think about it this way. When you're at the beach, right, and you're hanging out in the surf playing, you know, if you're out maybe like 100, 200 yards from shore, when the big waves come in, right, you kind of see them coming and you're just sort of like, whoa, you kind of go up and you go down, right? And then the other big wave comes, whoa, you go up and down, right? You kind of ride it out because the energy is all spread out in a much larger, wider wave. Whereas that same wave, by the time it reaches shore, the energy is compressed into a much more compact, tighter wave. And so if you're in the crash zone, that same wave can really roll you, right? And then you're trying to get up and you get knocked down by the next wave, right? So the energy, because it's compressed in a smaller wave length, actually represents more energy per, set of waves that are hitting you, right? So think about that. And this is where the whole particle thing comes in, because a photon then is a discrete packet of waves that are traveling together. So to use that beach analogy again, those of you who surf, right, you know all about being out there on your board waiting for a good set of waves to come in, right? And so the waves come in and they're in a set, and that's like a packet, right? A unit of waves that are coming in. And that's what a photon is, right? So a photon is a discrete packet or bundle of waves, but within that bundle of waves, the waves themselves can be really tight and compressed, high energy, or they could be more stretched out, right? Low energy, all right? So that's why light is considered both as a wave and as a packet. So this idea of a packet, this photon, is what we're talking about hitting, for example, a molecule of chlorophyll. Right? So that's why sunlight, the energy that's coming out of the sun is many different wavelengths. It's a huge range of wavelengths, and only the wavelengths between 400 and 700 are what we evolved here on Earth to be able to see with our eyes. I mean, you all know that other animals, other organisms are far more sensitive to like ultraviolet wavelengths or wavelengths that we cannot see, right? So sunlight contains all these packets, these discrete photons of different sized wavelengths, reds, greens, violets, yellows. And that's why when those packets go through something like a prism, right, not surprisingly, these different packets of energy will move differently through something like a prism. And so they end up, instead of being all together and moving in parallel, they get a little separated, right? A little deflected, depending upon their wavelength. And that's why you get a rainbow, right? Because then the different photons are coming out at different angles. And so you actually see them now as a nice range of colors. So when the photons hit the chlorophyll, what's going on, right? So packet of energy hits, let's say this carbon represented here in this chlorophyll molecule. And so here's the carbon. When the photon hits, it's going to disrupt the electrons that are in their clouds around the nucleus of the carbon atom. And what's going to happen? That energy gets put in, and it's going to cause those electrons right, to have more energy. So the electrons might jump to a higher sort of orbital, energy level, right? And as we've been learning, right, all semester long, electrons would much rather go back to their normal, happy, more comfortable energy level if possible, right? So in this case, jumping to another energy level represents the altering of double and single bonds, right? So these double bonds then will alternate and kind of flip around through this structure, right, as a result of photons repeatedly hitting this, okay? That's why this structure is so important for determining which wavelengths of light this molecule is most sensitive to. Think about it this way. The chlorophyll A and B will only get excited at certain wavelengths of light hitting them, right? And it's a function of its particular molecular structure. Let's imagine you have a glass full of water. I think you've all done something like this before, right? And if you had really loud volume music or something like that, right, you might actually see ripples forming in the water. Right? And so if you were to actually, let's say, have a whole range of bass sounds to treble sounds, and you were to sweep up and down, right, at loud volume, you might find that at certain frequencies, as you're moving down from the bass up through the mid-range, that's when you see the ripples appear in the water, right? And you get these kind of standing waves. But then as you take the frequencies higher, those ripples go away, even though the volume level of the sound, right, hasn't changed, right? But the wavelength of the sound has changed. So I think you all intuitively know that has to do with the shape of the glass, right? So depending on what shape container you're doing this in, right, you'd get the ripples at different frequencies of sound coming out of this loudspeaker here. Right? So therefore, the resonance frequency, the frequency where the energy is being absorbed and you actually can see it in the form of being absorbed by the water, has to do with the structure of the glass. Right? So that's what I'm saying up here. Right? The structure of the chlorophyll A and B is the right structure so that they are most sensitive to absorbing the green wavelengths of light. Right? And, oh, no, sorry, that's bad. They're most sensitive to absorbing the purple and the blue wavelengths of light, as well as the yellows, oranges, and reds, right? But they are the wrong structure to absorb the green wavelengths. So those are the wavelengths that bounce back. Right? Quick little fun point, right? If plants evolved with the sun shining and the sunlight has all frequencies of light in it, how come plants didn't also evolve chlorophyll molecules that could make use of the green wavelengths, right? I mean, that sounds like a pretty fair question, right? Why is it that they exclude or don't bother with the green, right? But they care about the purples, the yellows, oranges, and reds? Think about it this way, right? Throughout the course of a day, from morning to sunset, what colors do you see in the sky, right? Because those, when you look up at the sky, represent the wavelengths that are actually reaching your eye through the atmosphere. Right? You see plenty of things like blues and maybe even purples in the blue sky during the day. What do you see at the morning and at the evening? You see yellows, oranges, and reds. How often do you actually see a green sky, right? So in other words, as plants evolved on our planet in an atmosphere that was becoming increasingly filled with gases that filter out the sunlight and only allow or emphasize certain wavelengths reaching the surface of the planet, right? It just wasn't worth it for a chlorophyll to evolve to be sensitive to the green, right? Because you actually see very little green light reaching the surface of the planet. That's kind of fun to think about, right? Okay, so these big protein complexes then that are filled with chlorophyll and that are receiving the light, right? What's going on inside of them? So these complexes are what we call photosystems. Just like in the mitochondrial ETS, we said there's a complex 1, 2, 3, and 4, right, big Roman numerals. Here in the chloroplast, in the thylakoid membrane of the chloroplast of a plant, we have photosystems. And the three most important ones are photosystem 1, photosystem 2, right? And then there's this cytochrome B F complex in the middle. So that's shown in this simplified diagram here. You'll notice that photosystem 1 is actually the one on the right photosystem 2 is the one on the left here, right? So light is going to come down, strike those chlorophyll molecules. The double bonds of those chlorophyll molecules are going to start madly flipping around, right? Representing the energy kind of being rippling through all these bonds as the electrons are being excited and then relaxing again. Right? And that's going to power the stealing of electrons off of water, right? So ultimately, those electrons that are taken from the water are going to be passed through these photosystems, right? And it's going to be pumping protons across the membrane inside the chloroplast. And then those protons that are being pumped to this side, right? Huge gradient set up. Here it is, the protons on this side are allowed to flow back downhill through those ATP synthase pumps, right? So this part of it is just the same as what we learned before in the mitochondria. So how does the energy move through these different chlorophyll-containing proteins, right? You can think about it as this down here, right? This is an array of photovoltaic, solar cells, right, that are harnessing the sun's energy and turning them into electricity in this case, right? Notice how they are arranged, right, in these kind of like dishes, and you have many cells that can kind of help to focus the energy onto one point, right? So in here, the sunlight is all being focused by, actually, right, these probably aren't photosensitive cells themselves. These are probably just mirrors, right? But they're essentially bouncing and focusing the light to a very strong photosensitive cell in the middle that generates the electricity. So inside these photosystems, it's a similar type of arrangement. You'll have many proteins that contain or are hanging on to these chlorophyll prosthetic groups. So each one of these sort of discs here, these yellow discs, represent, maybe like a different pigment, a different chlorophyll that's being held on by a protein that's part of this photosystem. So when the photon hits one of them, right, it causes the double bonds, to kind of flip around inside of it. And importantly, as they're flipping around, the electrons in that molecule of chlorophyll themselves never leave the chlorophyll, right? They're just madly kind of flipping around, double bonds, single bonds, double bonds, single bonds. And if you have another chlorophyll molecule close to that, that crazy flipping around of bonds can actually be transferred. That energy can sort of bump the next chlorophyll molecule next to it, which is what this dotted red line represents, right? And so all the excited double bonds, single bonds in this chlorophyll sort of bumps into or shoves into, this chlorophyll, causing it to start flipping around, and so on and so on and so on. So we call this the resonance transfer, right? Because this is happening at the right frequency that these molecules are very sensitive to being able to be sort of disturbed like this, right? So that's sort of like this type of, in the 80s, it was really popular. We had places like Sharper Image. I don't even think we have Sharper Image anymore. That was a place in the mall you would go and buy all these like kind of fancy, executive desk toy type of things, like these balls, right? These kinetic balls. And you guys know how this works, right? If you pick this ball on the left and you sort of pull it up and let it go, it comes and it hits this one, and energy is transferred, that whole string of balls, right? And then the very one at the end gets booted off. So this part is the energy transfer part, right? You actually don't see any movement of these balls because the energy is just moving between them, right? So that's what the red dotted line is up here, right? The energy represented by all the single and double bonds flipping around inside these chlorophyll molecules gets transferred in a resonance form until it eventually reaches a special chlorophyll molecule, right, which we call the reaction center chlorophyll. And that one kind of is like the last ball on the chain down here, because that's the one where something actually happens, right? And in this case, the thing that happens is that resonance transfer energy actually causes electrons to get booted off of this special reaction center chlorophyll, right? And that electron then gets moved on and starts going through the chain, right? All right, so here's a more detailed figure of photosystem 2. Again, you have photons, packets of light striking the chlorophyll molecules that are being held as prosthetic groups by the different proteins that are making these big purple instead of big brown blobs, these look like purple bell peppers or something like that, right? And the resonance energy will then move through the different chlorophyll molecules, just like the way we had those solar dishes focusing, right, the energy. And in this case, they're focusing the energy onto this special reactive chlorophyll that we call the P680 chlorophyll, because it is most sensitive to energy from 680 nanometer wavelengths. Right? And what that does is it causes the P680 chlorophyll to cough up electrons. Right? And those coughed up electrons then are received by a electron carrying protein known as plastoquinone. Right? And so this plastoquinone, actually technically plastoquinone is not a protein, right? It's one of these other examples of a prosthetic root that can kind of like float around by itself, right? So here's the structure of plastoquinone here. Right? And so this QB then will move the electron, right, through the membrane over here. And in the process, right, because it's just in this case, right, it's an electron without a hydrogen, without a proton attached to it, right? And we all said before, electrons don't like to transfer that way, right? It's able to pull a proton from the stromal space, this side of the thylakoid membrane, and attach it to the electron, right? So that now the electron is being carried in a much more happy kind of condition, right? It gets moved to this cytochrome BF complex, where the proton gets removed, right? And that proton, when it's removed, it ends up being released on this side of the membrane. All right, so that's how the protons are being moved across from a low concentration to a high concentration, right? And that requires energy, right? Where does that energy come from? It comes originally from the sun, causing these electrons to be energized, causing electrons to be booted off the P160. So you said, wait a minute, I thought, Dr. Shen, you said that the electrons come from water. Yeah, they do. Because once this P680 chlorophyll gives up its electrons, it kind of needs to get electrons back, right? Because after a while, if it gives up all of its electrons, I mean, what happens to its chemical structure? So as the electrons are being booted off the P680 due to the resonance energy coming through the purple bell pepper here, right, through all the chlorophyll molecules, those electrons are replaced by the electrons that are taken off of the water, right? So that's why the water plays such an important role, because without the water here, you are not going to replenish the electrons on the chlorophylls that continuously get booted off, right? And that's why the water is so important as part of this first part of photosynthesis, and the taking of the pair of electrons off of the water, right, breaks it down into oxygen, which is released by the plant. Right. This is the last slide for today. And this is just some other cartoon ways of thinking about this, right? This is actually meant to kind of help you see it in slightly different ways that might make it easier for you to kind of visualize. So this is the photosystem, so massive structure of proteins with certain proteins just highlighted in green and whatever here. And what it's showing you, right, is that this is the chlorophyll that's being held on to, right? So remember, the chlorophyll is kind of like this fish net or bug net kind of thing, because here's that ring with the magnesium, and here's kind of the, I don't know, the part that you hang on to, right? So there's a couple of chlorophylls here, right? And then here is the, what is this? I think this is representative of where the water is going to come in and the oxygen is going to end up coming out. Right? So here you can see another view of it, that all of these individual complexes are protein subunits hanging on to chlorophyll, right? So each one of these is like when the sun hits it, it begins to agitate, the double bonds, single bonds begin to flip around in it. This one kind of crashes into this one, so that resonance energy is transferred through it. Right? And so you can see this is the path of the resonance energy through it, eventually leading to that central special chlorophyll down here that's actually going to manage the flow of electrons. Right? So here's the simple way of representing it. That's that special P680 chlorophyll in the middle that is going to receive all the focused resonance energy from all of the other chlorophyll containing proteins around it, right, causing electrons to be booted off of it that go to the plastic quinone and carried off into the electron transport chain. And importantly, this P680 needs to have those electrons replenished. And so those electrons come from the breaking of water, right? The breaking of water that then replenishes this P680 so that this process can just continuously keep going, just as long as there's energy coming in from the sun to power it. Okay, all right, let's go ahead and stop there.